Hi, and welcome to Chapter 21. In this chapter, we'll be looking at viruses, prions, and viroids. Viruses are really interesting because they are acellular. They're not composed of one or more cells, which technically makes them not living. They're non-living. They have no metabolism, they can't grow, they can't really do anything unless they are inside a host cell and hijacking, really using the host cell machinery, their enzymes, to complete these functions. We're not sure where they came from, although they seem to have picked up different DNA and RNA nucleic acids from different sources. They can infect pretty much everything. They infect humans, animals, plants, bacteria, pretty much anything you can think of. But they are species specific. For instance, if you have a dog or cat at home and they catch a cold and sneeze on you, it's unlikely that you're going to catch that particular cold virus because it's species specific. Viruses are usually also very tissue specific. For instance, HIV, a common example used here, human immunodeficiency virus that can lead to AIDS, acquired immunodeficiency syndrome. The virus can only be contracted if it enters certain cells in the body. So most cells of the body, HIV cannot enter, but certain cells found in our blood, HIV can enter. Viruses were known to exist before they were seen, and this is because they are super tiny. We cannot see them with the compound microscopes that we have in our laboratory. So the first virus discovered was the tobacco mosaic virus that causes plant disease in these tobacco plants. And filtration removed bacteria, but the disease was still present. So the people knew that something else was causing the disease. And it wasn't until the electron microscope was invented that we were able to visualize these viruses. So viruses are too small for us to be able to see them with the compound light microscopes we have in lab. The ones we have in our lab can allow us to see things like fungi or protozoa or even bacteria, and we're using the units micrometers, but we cannot see viruses. For viruses, we really have to use electron microscopes that allow us to see on the nanometer scale, so 10 to the minus 9 here. To kind of give you an idea of how small the viral genome is compared to something that's already really small, like E. coli. This is a bacterium. What they did here in this picture, or to create this picture, is they pulled out the entire circular chromosome. This is all just that single circular chromosome in E. coli, and they compare it with the viral genome, super small. So there are 4,000 genes on the chromosome for E. coli, but only 3 genes on this particular virus here. And for instance, HIV only has about 9 genes, and it can be deadly. So very, very small. So almost all viruses cannot be seen with our compound light microscopes. There are a few exceptions that we call giant viruses that we can kind of see with our light microscopes. And the reason we can't see most of them is because a single viral particle, which we call a virion, so this is a viral particle, as it is outside of the cell, can be only about 20 nanometers across in diameter. So having the electron microscope available really gave us insight into a whole new world of these viruses and their variations. Just to give you an example of their relative sizes, here's a eukaryotic cell, and we know eukaryotic cells are pretty big compared to prokaryotic cells like E. coli, which are 10 to 100 times smaller than eukaryotes. And then if I were just to zoom in, like on the surface of the prokaryotic cell, that's what you're looking at here. This is the prokaryotic cell, and right here we have something known as a bacteriophage. A bacteriophage, or phage for short, is a virus that infects bacterial cells. Very, very small. If we look at the basic shape or structure of the virus, known as virus morphology, the makeup of viruses include nucleic acid. So all viruses have either DNA or RNA, but not both. And the DNA and RNA can be double-stranded or single-stranded. So viruses are unique in that it is possible to have single-stranded DNA, and it's possible to have double-stranded RNA in viruses. Viruses also always have a protein shell known as a capsid. And in some viruses, this is not all viruses, but some viruses have a lipid envelope that surrounds the capsid shell. And the envelope is very similar to the phospholipid membrane that we see on our cell membranes. Viruses, when they're complete in their structure and they're found outside of the cell, again, those are called virions. So virions are when the structure of the virus is complete. There are many different shapes for viruses, but these four are pretty common. For viruses that infect humans and other animals, we often see icosahedral and envelope viruses. And for viruses that infect bacteria, we call them bacteriophage or phage for short. These often have this kind of spaceship-looking virus with a head and a tail, or filamentous viruses. Bacteriophage are very well studied, and in particular bacteriophage T4 that has this very characteristic head and tail shape. You can see them infecting a bacterial cell over here. Bacteriophage T4 in particular infects E. coli, and it has DNA as its nucleic acid and a capsid protein shell. In contrast, adenovirus is a virus that has DNA as its nucleic acid and again a capsid protein shell, but this one infects humans rather than bacteria. In fact, this one infects the human respiratory tract to cause cold-like symptoms. It does not have an envelope, so how it infects us is that there are certain glycoproteins, sugar protein complexes, that bind to certain receptors located in our respiratory tract. And then when they get into our cell, they inject their DNA and cause issues that way. There are many other types of non-envelope viruses. Non-envelope viruses are also sometimes called naked viruses when they don't have an envelope. And these include the virus that causes polio, papillomavirus, and hepatitis A, amongst many others. Here's another type of virus that has an envelope. So here I see some nucleic acid. I see the capsid protein shell that I saw earlier. But in this case, we also have an envelope, and the envelope is a lipid envelope derived from the host cell's phospholipid membrane. For this virus, this one happens to be HIV, human immunodeficiency virus, that causes AIDS, acquired immunodeficiency syndrome. And it also happens to be a retrovirus. Retroviruses are RNA viruses that contain their own enzyme, reverse transcriptase. So once they infect the human cells, they inject their RNA, then they use their own reverse transcriptase to convert RNA into DNA, and then the viral DNA secretly integrates itself into our own chromosome, so it hides in our DNA. So at its core, viruses contain DNA or RNA, but never both. And as I mentioned, it can be weird because you can have single-stranded DNA or double-stranded RNA. These nucleic acids can be circular or linear, and they can be in a single piece or in multiple segments or fragments. Overall, since viruses are very small, the genome is much smaller than a regular cell. And when it gets into the cell, when the viral DNA, or I should say, or RNA, it hijacks the host cell machinery so that instead of performing normal cell functions, the cell enzymes now focus on making new copies of the virus. For RNA viruses, the RNA that is injected can be transcribed, or it can be replicated to produce new RNA or more RNA. Unfortunately, RNA viruses during RNA replication, their enzymes make more errors in the replication process to almost spontaneously create new viruses through these mutations, through these errors. So as a result, RNA viruses mutate more frequently, which makes it really hard for us to fight them off. Every time we're finally recognizing the RNA virus, then it might mutate. It changes the protein shape on the capsid shell or on the envelope, making it harder for our immune system to attack the new version of the mutation. We have many different ways to group or classify viruses. Sometimes they're classified based on their type of nucleic acid they have or the function, to the type of capsid structure or overall shape they have, if they have an envelope or not, or the structure of their genome. As I mentioned, some viruses have an envelope, and this envelope is a lipid envelope that is derived from the host cell membrane during replication or release. So when the virus leaves the cell, it picks up some of that phospholipid membrane and it uses it to wrap around itself. So these envelopes are made of that phospholipid bilayer, but also has proteins attached to it. And these proteins are virally coded glycoproteins, sometimes known as spike proteins, that allow the virus to bind to the next host cell receptor. So these are important in host cell recognition. Without these proteins on the surface of the virus, they usually cannot bind to and enter the host cell. Interestingly, really interestingly, is when the virus is outside of the body, like let's say it's on the surface of a table of someone who has the flu and just sneezed. These viruses are pretty fragile and easy to destroy because they're just made of that phospholipid bilayer. Just using some soap or ethanol can easily break down that phospholipid bilayer, and now the virus will be missing those spike proteins that were embedded in that envelope, and now they can no longer bind to or enter a host cell. So envelope viruses are easier to destroy outside of the body compared to naked viruses, which have that capsid protein shell and no envelope, and their spike proteins are on the capsid protein shell. Proteins in general are harder to destroy compared to lipids like that phospholipid bilayer. Proteins are more stable, and to kind of think of a comparison, think of the proteins that make up your hair and nails. These are pretty rigid and pretty stable. Here's another example of the enveloped virus. You have some kind of nucleic acid, DNA or RNA, the capsid protein shell, and then here's that lipid envelope derived from the host cell membrane, and those spike proteins that are used to bind to the cell surface receptors of the next host cell. Once they bind, usually the virus can now enter and infect the cell. This is a real picture of the SARS virus using an electron microscope. And you can see those spike proteins, those glycoproteins, on the surface of the virus. Here I put a picture of an enveloped virus here next to a non-enveloped or naked virus. And again, naked viruses are harder to destroy because proteins are harder to destroy in general when they're outside of the body compared to lipid envelopes. So lipid envelopes are easily disrupted using things like soaps, detergents, ethanol, whereas proteins are much harder to break down using those same chemicals. Inside of the body, it's a completely different story because envelope viruses usually cause more persistent infections and more severe signs and symptoms of disease. So to give you an example of envelope versus naked virus, the flu virus, influenza virus that causes the flu, is an envelope virus. In contrast, the cold virus that causes the common cold is a naked virus or non-enveloped. And we know that inside of the body, a cold is generally less severe, causes less severe disease than the flu virus. This is something you're going to learn more in your next biology course, but just briefly, one of the things that non-enveloped or naked viruses often do is when they get into the cell and they make multiple copies of the virus, they eventually burst out of the cell, killing the host cell and releasing those virions, those particles, to infect later cells. So this is a rapid process. The host cell no longer sheds new viral particles since it's now dead. In contrast, envelope viruses do not generally cause lysis of the host cell. Rather, they bud out of the cell, leaving the host cell alive. And what that means is the host cell can continually shed viral particles, resulting in that persistent infection. Additionally, because the virus is taking up some of our own cell membrane to make its envelope, a lot of the times the envelope acts almost like an invisibility cloak for the virus. It might be harder for the host immune system to detect the virus if it's hidden or surrounded by the envelope, which is really the cell membrane. That takes us to the end of our first video for chapter 21. In our next video, we're going to look at how viruses get into our cells and cause infection.